It's time for another amazing chemistry video with Mr. Stapleton. Proudly sponsored by Farming Doing a Nice Coffee.
Hi guys, welcome to this video, which is a direct follow-on from the previous one. I've just cut it into two parts so that it's not too long. The first one I just went through with proteins. I went through primary structure, which is all about how the amino acids link within the protein itself. And then went through secondary structure, which was all about how the protein chain bonds to itself via the polar amide groups. So you get hydrogen bonding occurring between the oxygens and the hydrogen of the nitrogen amine group. Okay, so make sure you understand those before we come on to this one. This one today is going to be all about the tertiary structure. Okay, so this is obviously the third way that proteins bond. And the tertiary structure we're going to look at here is all to do with the side chains. Okay? So whereas primary structure was to do with the primary bonds occurred when we did that condensation reaction between two amino acids, and the secondary structure was all about the hydrogen bonds that are occurring between the two protein chains in the amide groups, the tertiary structure is all to do with the side chains. Now, in amino acids, there are 20 different R groups you can have. So there's lots of variation between the side chains and the type of bonding you can get. But basically, the three main types of bonding you can get in the tertiary structure is you're going to still have hydrogen bonding, which makes sense because we've got amine groups, carboxylic acid groups, hydroxyl groups from alcohols. Okay, you're then also going to get ion dipole interaction because you're going to have amines and carboxylic acids which can ionize, a bit like an amino acid does when in a switcher ion, which I went through in a previous video. And then you're also going to have a third type, which is actually non-polar covalent bonds. And there's only one amino acid that will allow you to have non-polar covalent bonds, and I'll explain about that shortly. Alright, so that'll be the third type we'll go through. But what I want to focus on as well in the tertiary structure is the effect that both pH and temperature have upon the tertiary structure of a protein. Okay? Because proteins, and when I'm particularly talking about proteins, I'm going to really specifically talk about enzymes. Enzymes are natural catalysts. Okay? So they're biological catalysts. They help you speed up reactions or processes inside our body. Now, they are very shape specific. Okay, so it's really important that you remember that, so I'm going to write it up top. Alright, and enzymes function is shape specific. So sometimes I hear this referred to as a bit of a lock and key process, where unless you've got the enzyme of the specific shape that you want, it's not going to fit in the way you want to speed up your reaction. And if you change the shape of that enzyme, you're no longer able to speed it up. So it's really important that your shape of your enzyme doesn't change. Now, pH and temperature do change it, and that's because of the way they influence these different groups. Okay, so these different types of bonding. So what I want to talk about and show you firstly is how these different types of bonding exist within the tertiary structure of a protein. So now what I'm going to actually do is, instead of drawing out the long carbon chain book like I've done before, I'm going to represent the protein chains just as like little bits of spaghetti like this. Alright, so we've got all these different protein strands. Okay. Now the protein strands can bond to each other in the secondary interaction via those hydrogen bonds. But then we've got some side chains that come off of these protein chains as well. And this is what I'm going to focus on with some of them. Okay, now some of those side chains have NH2 groups on them. Alright, some of those side chains have carboxylic acid groups on them. Alright, some of those side chains have hydroxyl groups on them. Okay, so we can get a lot of interaction occurring between these. What can happen is you can get some self-ionization occurring. So you can actually get the hydrogen here from the carboxylic acid group coming off the carboxylic acid and going over to the amine. So the amine now becomes NH3+. Okay. Now that you've got these ions in here, they can give you ion dipole interaction. So ion dipole interactions are quite strong. They require a decent amount of energy to break, so they help to hold the chain together quite strongly. You've got hydroxyl groups over here. Hydroxyl groups over here can be involved in hydrogen bonding Alright, hydrogen bonding, again, you know, is the strongest type of secondary bond. Alright, again, requires a decent amount of energy to break. So, you get hydrogen bonding, you get ion dipole interaction. Remember I said there's one particular amino acid which can give you non-polar covalent bonding. Now that amino acid is cysteine, okay, and cysteine has a sulfur hydrogen group on it as well. So it looks a bit like this at the end of the chain. So sulfur hydrogen, and then sulfur hydrogen like this. Now, if you manage to get two of these cysteine groups to line up opposite each other, what actually happens is this. Your hydrogens disappear, and sorry about the smudging, as I said, I'm trying some new pens, so in between it cleans off alright. And so what happens is that your sulfur from one cysteine group bonds to a sulfur from another cysteine group, and you get this non-polar covalent bond holding them together. Now this is actually what happens when you style your hair. So if you use water, when you're using water, you get these cysteine bonds to form. Right? However, because they're non-polar covalent, they're quite weak, so when your hair dries, quite often your style can disappear because the cysteine bond breaks, they're easy to pull apart. Okay? But that is the third type of bonding you can get within your protein. Now what we want to look at is what effect pH and temperature have upon these. So I'm going to start with pH firstly, and in particular what I'm going to look at is just the effect firstly of an increased pH. So I'll use the yellow for this one firstly. So in an increased pH means higher hydroxide concentration. Now hydroxide ion, alright, what do? They react or take H pluses. So obviously if you've got a higher pH, which is basic, it's going to leave your carboxyl group deprotonated. However, what it is going to do is it's going to strip the hydrogen off the NH2. So if that gets stripped off, what you've now got is just your NH2 group here. Okay. And now if that was involved in an ion dipole interaction, you've actually broken that ion dipole interaction. And so the parts of the chain that were being held together by that ion dipole interaction are no longer held together, and the chain will actually separate. Now when it separates, what that does is that changes the shape, and as we know, enzymes are shape specific. Alright, so if you change the shape of the molecule of the enzyme, alright, it no longer is able to perform its biological function, and that term is denatured. Alright, so it's really important you know that term. The enzyme isn't killed. A lot of people think that enzymes are killed when they're denatured. They're not. It's just their shape changes, which means they can no longer perform their biological function. So basically, they're useless. We can't use them. If we go through and we decrease the pH on the other hand, so if we decrease the pH on the other hand, what we've got is a higher concentration of H+. So now what that H+ does is it comes in and it reprotonates the amine. So we get our NH3 back. However, what it also does now is it protonates the carboxylic acid. So again, a lower pH now breaks again the ion dipole interactions that we had before, which changes the shape of our enzyme, which means it's denatured, which means it can no longer perform its biological function. So that's really, really important to understand about the effect that higher and lower pHs have upon the structure, in particular tertiary structure of our protein, by either protonating or deprotonating the amine, or protonating or deprotonating the carboxylic acid. Now the other thing that can influence is temperature, and temperature comes in between these non-polar covalent bonds and our hydrogen bonding as well. Okay. What you're doing when you're supplying energy, and so in particular what we're talking about is increasing the energy. So if we increase the energy, what we do is we cause the particle to vibrate faster. What that can do eventually is cause these sulfur-sulfur non-polar covalent bonds to break, or our hydrogen bonds to break, because we're supplying energy. As soon as we break the bonds, the shape of the enzyme changes, which again means it's denatured. Okay? If you decrease the temperature of the enzyme, it can have the same sort of effect as well, of that you actually collapse the protein in on itself, right, because there's not as much flexibility within the bond, and so again, you're changing the shape of it, and so it doesn't work very well. What you will find is that for all enzymes, you will normally see a graph, okay, that it looks a little bit like this. I'm just going to shift over a little bit. So you're going to see a graph that looks like this. Alright, and you're going to have say pH along the bottom here, and you're going to have rate over on this side. And what you'll find is that most of them tend to look a little bit like this. And there's a certain pH here where it is most effective, and either side the rate of the reaction decreases because the enzyme obviously changes its shape, and so it becomes denatured at a certain point and won't perform its biological function. You can do exactly the same with temperature. Right? Temperature is a little bit different, okay, because of the fact that you do completely denature your enzymes at a higher temperature normally. Temperature should be down here, and rate should be up here again. And what you find is that generally as you increase your temperature, you get to an optimum temperature like this, and then your rate will just go bang, like that. Okay, so you've got an optimum temperature around here, and after that your enzyme becomes completely denatured and it will no longer increase the rate of a reaction. And generally once it's denatured you can't go backwards, alright, so you've lost the ability to be able to catalyse your reaction. So there's a lot of stuff I've been through here, alright, you can tell that I've only got a very small space for my head, so I'm going to stick in this spot right here. What I encourage you to do is make sure you can go through and understand the primary, secondary and tertiary structures of the protein, what influences that. What you really need to take out of this one is the different types of bonding that occurs, and in particular how pH and temperature changes really influence your tertiary structure. Alright, so your pH will influence your ion dipole interactions in particular. Right? Can influence your hydrogen bonding as well if you've got hydrogen bonding off of just a carboxylic acid group, but most of the time that will have self-ionized with an amine group, so it'll be in the carboxylate ion form. And so it's mostly your ion dipole interactions which are affected by pH. Either an increase in pH, which will deprotonate your amine group, alright, and so you won't be able to have your ion dipole interaction, or a low pH, which will protonate your carboxylic acid group, and so again interfere with your ion dipole interactions. And temperature for both hydrogen bonding and non-polar covalent bonding, what that does is that breaks apart your bonds, changes the shape of the molecule. Because enzymes are shape specific, if you change the shape they can no longer perform their biological function and they're what we call denatured. So hopefully this has helped you out. If you have any questions, as always, just ask. Otherwise, good luck. Thanks guys.